Okay, in this lecture video, we're going to move one step up from our lipid molecules and start talking about membranes and transport across membranes. So we've already learned in our in-class activity that we talked about, based on the structure of lipid molecules, we know that lipids aggregate together to form different types of larger structures. And one way they can aggregate when the area, the surface area of the polar head group is similar to the surface area of the nonpolar tail group, you can get these cylinder shapes of polarity, and we know that those can aggregate nicely like this to form a membrane. And that membrane is a bilayer, so we've got two of them. And what is the purpose of this? Well, lipids have a really big job when they aggregate to form our cell membranes. So sort of a two-pronged purpose. The first is that it functions to separate cellular components. So not only does it give us our cells, but it gives us all the organelles within our cells. It keeps everything in the right place. And in that sense, cell membranes serve as the gatekeepers. So they modulate which molecules can come in and which molecules can exit. And we saw already, like for example with the post activity, that you can change the structural integrity of a cell membrane, for example with the phospholipase, and that can make it possible for contents for that membrane to get leaky. That's a problem. You want your membrane to be structurally secure so that it's the proteins within the membrane that can then modulate molecule entry and exit. So this function then is going to depend not only on the lipids that are present, but also on the proteins, because our cell membrane is not just made out of lipids. 30%, in fact, of the human genome is made out of membranes, or sorry, not membranes, is made out of lipid. That's not right either. 30% of the human genome is made out of proteins that are interacting with lipids within the cell membrane. So let's first look at the organization of that cell membrane, and it follows something called the fluid mosaic model, which looks like this, that I stole off the internet. For some reason, the little website didn't come along with the picture. Sorry about that. The picture I tried to take out of your book kept giving me glare because I'm sitting under a really bright light. So I had to go with plan B, which was find a good pic on the internet. So here's the fluid mosaic model. All the pictures look the same. And you saw this when we watched the video, Inner Life of the Cell. So we have, first of all, this bilayer structure, so two leaflets, and we know that we have hydrophobicness, so the hydrophobic tails are within the middle of the cell membrane, this bilayer, they're pointing in, and the hydrophilic head groups are on the outer and inner sides, then, of those leaflets. So that's how we're protecting those hydrophobic tails from exposure to water, right? And we know that that's the driving force for the aggregation of lipids in the first place. It takes us all the way back to the entropy of water as being the driving force for that process. So you've got the hydrophobic effect driving that process forward, and you've got those van der Waals interactions from the interactions between the hydrophobic tail groups. Okay, so that's the first part. We have a bilayer structure. The second part for the organization of this fluid mosaic model is that this membrane is impermeable to polar or charged solutes. So molecules that are polar or charged cannot pass through that membrane. And we can see that by looking at the energetic changes that would take place for that. So let's see if I can take a picture here of my transparency without getting too much glare. Alrighty, I think that will serve my purpose. Just ignore the big glare spot there. So this is a diagram of free energy versus moving, essentially, a charged or polar solute across the membrane. So up here in part A, we've got this polar solute that's hydrated. It's surrounded by water molecules. So process-wise, in order to make it pass through that cell membrane, you've got to rip all of those water molecules off, pass that charged solute through the middle of the nonpolar bilayer, and then rehydrate those charged molecules, that polar solute with the water molecules. So you can look at how that free energy change takes place. That's shown here in red in this picture. So we know that in terms of desolvating and resolvating the solute with the water molecules, that's not that big of a deal. We're breaking those bonds, but then we're putting them back. But you can see there's this large activation barrier in order to make that happen. And then you can see that during this process that we're passing that that polar solute through the nonpolar bilayer, we're at a really high energy. So overall, the delta G double dagger, the free energy is associated with that activation barrier, the activation energy, is pretty high. So that's where our transporter molecules are going to come in, and we're going to talk a lot about those. So now you can see that we still have this process where we have to desolvate our polar solute, so down here in part B, and then resolvate it at the end. But now we've got a protein molecule that's lined with hydrophilic amino acids in the middle that can interact with our polar solute, so that's a good thing. So now we've got, essentially we're desolvating, but then replacing those water solute interactions with solute protein interactions. So it's not that big of a deal. We have to take the water molecules off, but we're replacing it with a new interaction with our protein transporter. So we're going to talk a lot about how these membrane proteins serve that purpose. Okay, so back to our description then. So we've got so far the fluid mosaic model. We have a bilayer structure. We have that it's impermeable to polar or charged solutes. The third thing that you saw in inner life of the cell, I want to write in yellow. The third thing is that molecules can undergo lateral diffusion. So that means within their individual leaflet, they can move laterally. So that means they can stay within the plane of their leaflet. And there was a really cool experiment done to show that molecules were free to laterally diffuse. So each leaflet, each layer of that bilayer is mobile, sort of like lilies on a pond. So the experiment they did to figure that out was back in 1977. It was an experiment by Michael Edidin from Johns Hopkins University, and he did immunofluorescence. So immunofluorescence means he's taken advantage, fluorescence, of immune system molecules. So he's using antibodies, and those antibodies fluoresce. They are tagged with a fluorophore. So what he did was he took two different antibodies. So he took some antibodies that recognized some proteins on the cell on a mouse cell. And that particular antibody was tagged to a fluorophore that fluoresced green. And it's funny, if you saw on the chalkboard on the floor, on the third floor, there's a picture of a round circle, and it says that this is the chemist's view of a cell, and it's a biologist's view of an atom. I thought that was hilarious, because it's true, we all draw them like that. So he took, back to the story, so he took these antibodies that reacted with mouse cell proteins, and when they did that, those antibodies fluoresced green. Then he took a human cell, and he had some antibodies, and those antibodies recognized proteins on the surface of the human cell, and when they bound, they fluoresced red. So he took these two test tubes of cells with their antibodies, and he mixed them together in the presence of the Sendai virus. And that virus has the ability to fuse cells together. So what happened, you can show this as a function of time. So you've got your mouse cell over here with its green antibodies, and you've got your human cell over here bound to its red antibodies. And what he saw was over time, so first those two cells fused. But then if you waited a while, and he waited about 40 minutes, what he found was that now you saw fluorescence of both antibodies throughout the cell. So those antibodies had mixed. And then what he found was that if he dropped the temperature, so if you lower the temperature from 37 degrees Celsius to 15 degrees Celsius, he slowed that diffusion down. So it took longer for that mixing to occur, which was the second experiment to really confirm that you had this lateral diffusion of molecules. Okay, so that was a really cool experiment. Okay, so the fourth thing then to talk about with our fluid mosaic model, or the fourth property, is that leaflet flip-flop is rare. Okay? So it's really difficult, for example, to take a lipid molecule from this leaflet and have it flip to this leaflet. And you've already talked about that as part of your activity we did with our lipid molecules, right? Because in order to do that, you've got to take that polar head group and pass it through the middle of the bilayer. Now, it's not that that can't happen ever. It can, but it requires assistance from a protein called a flipase, right, which will basically protect that polar head group as it passes through the center of the bilayer. So that tells us then that the distribution of components, so what you have on each leaflet of this particular membrane, can be asymmetrical. So the organism is very, very picky about which lipids it has where, which proteins it has where, and you can see there's a huge mix of everything here. So we've got lipid molecules, we've got protein molecules that pass all the way through the bilayer, we've got lipids that are glycolipids, so they have sugars attached, we've got proteins that we'll find on one side of the layer and not others, and we're going to go through all of that. So something that's true about this particular, about a membrane or a bilayer structure is its fluidity depends on temperature. We've already taken a look at this again in that activity you did in class with the lipids, because the temperature is going to affect the strength of the intermolecular forces between the lipids. The stronger the forces, the more solid-like packing that you will get. The molecules will be closer together and more rigid. So the temperature, well, you can basically have a temperature where you have really restricted motion for your bilayer, and we would represent that like that, where we're showing these nonpolar tails sort of in a rigid picture. And then you reach the transition temperature, which is called TM, the transition temperature, or the melting temperature of that membrane. So you hit a point then where these tails become really fluid. So that's why we always draw them with little wavy lines. So that means these tails are swinging all over the place. And what that transition temperature depends on is it depends on the chain length, right? So the longer the fatty acyl tail, the more van der Waals interactions you'll have between those tail groups. So it will take more heat to break those interactions up. And it also depends on the degree of saturation. So the more double bonds you have in those fatty acids, the more kinks in the hydrophobic tails, the easier it is to break them up. So just to get an idea for that, if you take a fat, standard nomenclature 14-0 versus 16-0, so no double bonds, and you look at the transition temperature, it's 24 degrees Celsius. If you add just 2 more carbons, it goes all the way up to 42 degrees Celsius. And that's not even changing the degree of unsaturation. So the melting temperature, the temperature at which your bilayer goes from a more solid structure to a more fluid structure, and that fluid mosaic model really comes into play in terms of diffusion, is very sensitive to how many carbons you have and the degree of saturation. We also saw in our activity the other day the effect of sterols, so looking at cholesterol's effect on the melting temperature or the fluidity of your bilayer. And we saw two different effects. So we saw that below the TM, so if you remember, this was the temperature, this was the fluidity, and in the absence of cholesterol, the change in fluidity as a function of temperature looked like that. So this point right here, that would have been the melting temperature, right? It hits a temperature and it immediately changes its fluidity. It goes from being more solid to instead more fluid. So that's what we were looking at with that graph. But we saw that in the presence of cholesterol, we had a more linear change in fluidity. But you could see from this graph that at low temperatures, cholesterol, fluid, the presence of the membrane and the fluidity of the membrane in the presence of cholesterol was more fluid because this line is greater than the membrane fluidity in the absence of cholesterol. But at high temperatures, cholesterol reduced membrane fluidity. So it was below what it was in the absence of cholesterol. So you have to think about what's going on there. So you could think structurally below the transition temperature, when you add those rigid rings from those four planar fused rings of cholesterol, those rigid rings disrupt the packing between the lipid molecules because you're not going to get as good an interaction with those planar rings as you are, for example, between a whole bunch of saturated fat. So therefore, that would increase the fluidity of the cell membrane. If you are above the TM, on the other hand, you could think that those rigid rings instead now will interfere with the lipid tail motion, right? So if fluidity has to do with the movement of those lipid tails all over the place, and you stick in a rigid set of rings, they're going to be bumping up against it. So that instead will decrease the fluidity of the membrane because it's restricting the space with which those tails can move around. Okay, so in our next lecture video, we're going to talk about some membrane proteins.